It is. Welcome everyone to our first Tubman Talk of 2025. We are really excited to welcome Dr. Goodridge today. I'm Dr. Bianca Beauchemin. I'm an assistant professor in the School of Gender, Sexuality, and Women's Studies, and also a member of the executive committee here at the Tubman Institute and a co-coordinators of the Tubman Talks. Now, Tubman Talks, as you know, if you've come here before, but if you're new, welcome. It's a space where and a platform to share research and publication with audiences who are doing work related to or interested in Africa and African diaspora. The Tubman community is interdisciplinary, and we welcome a wide range of topics and backgrounds. Tubman Talks are intended to provide an opportunity for individual scholars to critically strengthen their own research by presenting it to others in the spirit of engaging in debate and discussion. Before we get started, I would also like to begin with a land acknowledgement. I wanted to acknowledge our presence on the traditional territory of many Indigenous nations. The area known as Tekaronto has been caretaken by the Anishinaabeg Nation, the Haudenosaunee Confederacy, and the Huron-Wendat. We acknowledge the current treaty holders, the Mississaugas of the Credit of First Nation. This territory is governed by the Dish with One Spoon Wampum Covenant, an agreement to peacefully share and care for the Great Lakes region. In making this land acknowledgement, I honor and acknowledge the land and its people who have been here for thousands of years. In relation, I would be remiss not to mention the current genocide happening in Gaza, the catastrophic situations in Sudan, Congo, and Haiti. It is important to contextualize the repressive colonial and imperial oppressions that undergirds the current situation. Influenced by my personal Black feminist politics, scholarship, and praxis, I recognize that my own political commitment mandates a solidarity that is bound up and intertwined with the liberation and self-determination of Indigenous people, as well as Palestinian, Sudanese, Congolese, and Haitian people across the world. So for us today, we have a great treat. We have the talk by Dr. Lawrence Goodridge entitled One Health Approaches to Health Equity. Dr. Lawrence Goodridge is housed in the Canadian Research Institute for Food Safety, where he holds the Canada Research Chair in Foodborne Pathogen Dynamics. Dr. Goodridge conducts research related to control, detection, and surveillance of pathogenic microorganisms, including bacteria, viruses, and parasites. Dr. Goodridge has published more than 120 peer-reviewed journals, articles, which is incredibly impressive, and book chapters. Has been awarded more than 50 millions in research funding from US, Canadian, and international funding sources, and is regularly interviewed by national and international media on topics related to infectious diseases. After completing a Bachelor of Science with honors degree in microbiology at the University of Guelph, Dr. Goodridge completed both his his Master's of Science and his PhD degrees in food sciences with an emphasis on microbial detection of foodborne pathogens, also at Guelph, before leaving for a postdoctoral fellowship to continue his training in microbial food safety in the laboratory of Dr. Michael Doyle in the Center for Food Safety at the University of Georgia. Prior to returning to Guelph, Lawrence has held faculty position at the University of Wyoming and Colorado State University. More recently, he has the Ian and Jayne Munro Chair in Food Safety in the Food Science Department at McGill University. And finally, in January 2019, Lawrence joined the Department of Food Science as a director of the Canadian Research Institute for Food Safety. So today for the talk, One Health Approaches to Health Equity, is an engaging talk at the Harriet Tubman Institute, which will happen after I finish talking. Dr. Lawrence Goodridge is an expert in food safety and public health microbiology and the Canadian Research Chair in Foodborne Pathogen Dynamics. He will delve into the transformative role of wastewater surveillance within the framework of One Health Approaches to Health Equity. The One Health paradigm emphasizes the interconnectedness of human, animals, and environmental health. Dr. Goodridge will illustrate how wastewater surveillance as a powerful epidemiological tool aligns with his approach to address pressing health disparities in diverse communities. Drawing from his extensive research, Dr. Goodridge will explore the potential of wastewater monitoring to detect emerging infectious diseases, track microbial resistance, and assess public health trends with precision. This non-invasive method provides timely community-wide insights, enabling public health officials to intervene proactively, particularly in underserved and marginalized population, often disproportionately affected by health crises. By monitoring biomarkers and microbial indicators in wastewaters, this approach bridges gap in traditional surveillance systems, offering a cost-effective, scalable solution to promoting health equity. The presentation will highlight key studies during key case studies from Dr. Goodridge's work, including the role of wastewater surveillance during the COVID-19 pandemic and its application in monitoring environmental contaminants. He will also discuss the ethical consideration and logistical challenges inherent to implementing this technology on a global scale. Ultimately, the talk will emphasize how wastewater surveillance embodied the One Health vision, fostering resilience and equity in public health systems across the globe. Attendees will gain a deeper understanding of the potential for wastewater surveillance to transform public health practices, ensuring equitable access to health intervention and safer futures for all. Please now take the time to welcome to Tubman Talk, Dr. Lawrence Goodridge.
Thank you very much. Can you hear me?
Yes.
Okay. Good afternoon, everyone. I'm just going to get my slides up here. Okay. So good afternoon, everyone. Thank you so much for inviting me to be a speaker, the first speaker of 2025. And so today, what I would like to do is just present some of my research related to One Health approaches to health equity. And we'll be talking, as we heard in the abstract, on wastewater-based epidemiology. And so before I begin, it's important to understand what One Health is. And so One Health is an approach. It's an emerging approach where the idea is that we understand that certainly with respect to diseases, and we'll be focusing on infectious diseases, that these diseases can be transmitted from animals to humans through the environment. And traditionally, research on such diseases was conducted in silos. That is, researchers would work on animals, perhaps veterinarians would work on the animals, those in the human medical profession would work on the human side of diseases, and then there were environmental researchers that would, study how these diseases or contaminants are spread in the environment. But we learned that in order to really make strides towards eradication of disease, or at least a decrease in the disease rates, we really need to stop working in silos and work together. And so that is what One Health is. It's focused on studying contaminants and disease in humans, animals, and environments, but in an integrated approach. And so that's what we're going to talk about today, because the diseases that I'm going to talk about today, emphasis on foodborne diseases, as well as COVID-19, we know originate in animals and have been transmitted to humans, and as we'll see, through the environment. And also, surveillance of wastewater as an environmental source is a great way to assess the presence of these contaminants. So, I also include in my title comments related to health equity. And that's because we know that health inequity is a major problem globally, and certainly in Canada. And so when we talk about health inequality, we're talking about disparities in health outcomes and access to care across different populations. Now, certainly in developed countries, we see that these populations tend to be those that are marginalized, and have lower socioeconomic status. And so this means that social determinants of health inequality are important. And we see some here. This is not exhaustive by any means, but certainly income, education level, housing, geography, and systemic racism are all social determinants of health inequality, and have all been linked to increasing levels of disease in populations that are marginalized. And certainly, as we see the social fabric in Canada begin to disintegrate, higher numbers of homeless people, more people living together in close populations, less access to health care, we can begin to see increases in disease. And so, because of that, marginalized communities experience higher rates of preventable diseases and lower life expectancy than those in majority communities. And so, this graph here, this is from the Public Health Agency of Canada, and what it shows is the life expectancy at birth by the material and social deprivation index, which is a composite indicator used to measure and analyze the levels of deprivation within a population. Okay, and so this combines two distinct dimensions of deprivation: material deprivation, which refers to a lack of access to basic resources and goods that are necessary for maintaining a standard quality of life, and social deprivation, which reflects challenges in social inclusion and community connectivity. And so what we see is that if you look at the top, so we see men, women, and then both sexes at the top. And if you look at both sexes at the top, the life expectancy between those that are least deprived and those that are most deprived is almost 6 years. Okay, so what that means is that the people that are most deprived materially and socially live on average 6 years less than those that are least deprived. And so, we can begin to see how health inequality in Canada not only reflects one's day-to-day life, but also their complete life expectancy. And then when we move forward to infection and focus, exclusively on infectious disease surveillance and inequality, we see several challenges here. We see limited access to testing and treatment in rural communities and in indigenous communities. And really, when we talk about indigenous communities, what we're talking about are people that live in closed communities. So that would be First Nations, for example, that tend to live in communities, including remote communities, Inuit communities, for example, which we saw and we'll take a look at with respect to COVID. We saw these communities largely um, limited in terms of access to testing and surveillance and treatments and so forth. There are several reasons for that: language, infrastructure, and systemic discrimination. And so, we often see when it comes to infectious diseases underreporting of diseases in underserved populations. And as I said, one example of that, recent example, is the unequal COVID-19 testing rates that were observed in urban versus remote areas. And I just want to focus on COVID-19 for a few moments here, because I really think this shows the issues that we're facing when it comes to health inequality. So what you're seeing are maps. This is of the city of Toronto, or well, of Toronto and surrounding cities, including Etobicoke, North York, and Scarborough. And the map on the left represents the raw case numbers. So that's the purple color, the purple polygons. This is the raw case numbers of COVID-19 and the total population in each neighborhood. Okay, and so you can see that the first thing you can see is that there is a correlation. This is a graph on the left. There is a correlation between a positive correlation between the higher population and the more cases. Okay, so if we look, for example, on the graph on the left, if we look at the far left, Etobicoke, and the top, the dark purple there, just above Etobicoke, you can see large population, large number of COVID cases. Same thing if we look to the far right, to the right above Scarborough, large number of people, um, and high cases. If you look right below where it says Toronto, the same thing. Okay, so this makes sense. COVID-19 highly infectious. The more people that are present in close proximity, the faster the disease will spread. On the right side, the graph on the right shows the average annual household income, which is shown by the blue circles, as well as the COVID case rate in that neighborhood. And so here we begin to see some differences as it relates to health inequality and social determinants of health. So you still see, and the darker the color, the darker the red color, um, the higher the cases. So again, we see higher cases in Etobicoke and Scarborough, for example, where income, average annual household income is lower. Conversely, if you look um, in the Toronto area, and so above where it says Toronto, see a big blue dot there, very high household income, very little COVID. Uh, and then also, if we compare the average household size with the COVID-19 cases per 100,000 people, we see that in many cases, the larger average household size, the more cases. So that is, could be for several reasons. First, the fact that there are more people in physical close interaction with each other daily, and the disease is highly infectious, so it could spread. But it's also true that there are larger household numbers, or people living in households or in houses in neighborhoods with lower socioeconomic status, because people cannot afford to live in their own houses, so they tend to congregate. That also likely means that people in those households are probably working in jobs that expose them to higher risk of getting COVID-19. So service industry, like for example, the food industry and so forth, which we know during the pandemic, many people in those types of industries had much higher rates of illness. They had to work or else they wouldn't be paid. And those industries were really the backbone of, 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 of the country during that pandemic, at least during the years when the country was largely shut down. So this really shows how social determinants of health can affect health inequality, and how that can lead to higher rates of disease and even death among those that are marginalized. So what can we do about this? Well, one of the issues that we have in Canada and other developed countries is the approach that we use to conduct surveillance. And this is surveillance for any disease. It could be an infectious disease, it could be a non-infectious disease, like cancer, for example. And this is what we see here. It's called the pyramid of surveillance. And so at the bottom, we have exposures in the general population. So let's say we're talking, and in this particular example here, you can see the arrow that says average 3 to 4 weeks. So this is a particular example as it relates to foodborne illness, but this can work for any illness. But let's talk about foodborne illnesses. So at the bottom of the pyramid, we see exposures in the general population. So let's say all of us here today went to a restaurant after this lecture, and we ate. And the food at that restaurant was contaminated with a foodborne pathogen. So we're all exposed. Not every one of us will get sick. Only some of us will get sick. So that's the second rung on the pyramid. That's called the attack rate. For different pathogens, there's different attack rates. The point is that not everybody will get sick, only some. Let's say 75% of us that ate at the restaurant get sick. Now, if I'm going to assume that almost everyone here has had a foodborne illness. And if you have, I would hazard a guess that you did not go to the hospital, or at least most of you who had a foodborne illness did not go to the hospital. Most people who get sick, the first thing is you don't want to go anywhere except perhaps the bathroom. And in 24 hours to 48 hours, you start to feel better, so you don't go to the hospital. So that right there is the main issue. If we don't go to the hospital, if we don't seek a medical profession's help, then public health officials will never know that we're sick. So of the people who become ill, some people will seek care, but the vast majority, at least very early in the outbreak, will not. If one does go to a hospital or a public or medical professional, they have to take a specimen, typically a stool culture, and that is used to test for the agent that's causing the disease. But in some cases, doctors don't do that. They say it sounds like you have a foodborne illness. Go home, drink some fluids. If in 48 hours you don't feel better, come back. So if a specimen is never obtained, public health officials will never know about the issue. Even when a specimen is obtained, it has to test positive for the agent causing the test. No test is infallible. So for various reasons, the test may not yield that organism. Maybe by the time you go to the doctor, it's not present in the stool, or the test failed, or whatever the case may be. So only when the test tests positive for the agent is that called a laboratory confirmed case, at which point it needs to be reported to the health department. Now, during the COVID-19 pandemic, with respect to foodborne illnesses, that didn't happen very much. And the reason it didn't happen was because the health professionals were worried about more serious things, such as COVID-19. So, because of this, it takes an average of 3 to 4 weeks from the time someone consumes a contaminated food to the time that health professionals know about it. All the while, that food is still in circulation. It's in grocery stores. People could be going into grocery stores and buying the food, and consuming it. And so the other main thing is that we don't know. If you look at the pyramid at the top there, the number of cases reported to the health department as compared to the actual number of people who became ill at the bottom of the pyramid, much smaller. So that it means we don't know about most people who become sick. And if we don't know about most people who become sick, then there's very little we can do about those people. And so one of the things we need to do is to move from this surveillance approach, which is passive in nature because it requires a sick individual to initiate and seek the help. We really need to move to an active surveillance program. Moving to an active surveillance program has several obvious advantages. In addition to everything I just told you, from the point of view of health inequality, we know that certainly people who are on the lower socioeconomic status are even less likely to go to the hospital. They don't have time. If they need to take time off work, they're not going to be paid. So it's likely that those are people, if we were to stratify this by socioeconomic status, those that are most marginalized are likely among the least to go to the hospital. So if we were to adapt an active surveillance system that moved away from relying on the sick individual to initiate the surveillance, this would also help with health inequality. And so this brings me to wastewater-based epidemiology. So wastewater-based epidemiology is, I was going to say it's an emerging field, but it's really not. It's been around for hundreds of years. And what it really refers to is that infectious agents will be spread in fecal waste. And, given the way our cities are set up, that fecal waste will be transported to a centralized facility, allowing one to sample an entire population from a single waste facility. So it's cheap, it's non-invasive, um, and and one can gain a lot of information regarding infectious diseases and so forth from it. Generally speaking, privacy, there's people always ask me, well, what about privacy? Um, so generally speaking, there are very little privacy issues because it's a mixed sample. The caveat being the size of the population. So, we can sample, the city of Toronto has like 4 or 5 wastewater treatment plants. Guelph has one, for 120,000 people. So if we sample at that level, privacy issues are non-existent. However, we can sample, we can sample in manholes on a street. If we were to do that, then the population we're sampling is much less. It's the population of that street. And so let's say we knew that on that street, somebody was living there that had a prior conviction for drugs. And we were sampling and we found a drug, illicit drug, in that wastewater. We might conclude that it was from that person that we have the additional knowledge. So there are some privacy concerns related to the size of the community. For example, if we were sampling in a remote indigenous community, that's another concern, where the community is closed. There's only a certain amount of people, so if we find something, that could be associated with privacy issues. So there does need to be some policy that's developed around that. But for what we're talking about today, we're talking about sampling at population at city level, or populations within a city that are so large that such concerns are minimized. And so, really, the story of wastewater begins in the 19th century in London, England, where John Snow is the father, widely considered the father of epidemiology. He was the first to be able to show how diseases like cholera spread. And in that case, a major outbreak, it spread through water pumps, such as the pump in the picture. This is it's called the famous John Snow water pump. And I was there, and anybody who does work in epidemiology, it's kind of like a pilgrimage. You go and see this water pump because John Snow is the one who really started the field of epidemiology. So I was there about 10 years ago in London, and so forth. And so he was able to show that people congregated at these water pumps, and this is how the disease spread. So what he did, and at the time, the politicians did not believe him, and they wouldn't listen. So what he did was he removed the handles from the pump, so people couldn't pump water, and the outbreak ceased. And so today we now see that as a huge, um, a seminal moment in epidemiology. In the 1900s, we see early monitoring for pathogens in sewage systems. Again, pathogens are spread through fecal waste, so sewage and this is why we need to wash our hands after going to the bathroom and so forth. So sewage is a great way. And the idea was to understand how waste reflects population health. And really, that's really the goal of wastewater-based epidemiology today. Fast forward to the 2000s, and we see the use to monitor illicit drug metabolites in wastewater. And that's still happening today. Cannabis, for example. Statistics Canada monitors cannabis use for cannabis metabolites in wastewater in different cities to gain an understanding of the usage. Between, a big gap, which I didn't fill in because of the time limits that we have, but I can tell you that really between the 1950s and the 2000s, wastewater surveillance was really used for polio, and also uptake of the polio vaccine. Okay, and then so 2000s we see drug metabolites, and then we come to 2020 and COVID-19. And early on in the pandemic, it was reported that one of the main symptoms of people who had COVID-19 was diarrhea. And so this led researchers, I think in China, where COVID-19 originated, to look at the wastewater. And sure enough, they found the virus in wastewater. And so this really led to an explosion in wastewater-based epidemiology to track COVID. And so that's happened, and I have to say it was met with degrees of skepticism. Certainly here in Canada. In Canada, we were among the last to adopt this. and there was a lot of skepticism. But really, during after 2022, when clinical testing largely stopped, it's certainly in Ontario, where we had a robust program, which we'll talk about later. Wastewater testing was the only way that we knew that there were waves. So if you remember, and you might have remembered back to seeing on the news when they would say, "Oh, we're entering a wave," that data all came from wastewater testing. It was really the only way that we could accurately track it. And now it's used to track other issues related to public health, antibiotic resistance, which has been referred to as a silent pandemic. 15 people in Canada die every day due to antibiotic resistant infections. And it's thought that by the year 2050, globally, as much as 39 million people will die every year because of this, which is more than diseases like cancer and other diseases. And so wastewater is now, surveillance is now used in over 50 countries. Um, with respect to COVID, this really shows why and how it can be used. So this is a map of the world, and all those lines are and the circles are major international airports. And you know, what this shows is the spread of SARS-CoV-2 and the different variants of SARS-CoV-2 across the continents as overlaid on the flight routes. And so basically, everywhere that, where, um, you know, there was flights, is COVID spread, which is why there was an attempt, which didn't really work, but the attempt to shut down flights. And so one of the things being proposed currently is a global wastewater monitoring um, program at the world's busiest airports. Okay, because this is where people come in. And so certainly in a country like Canada, where pandemics are not likely to begin, but we will be a recipient of that, monitoring at like Pearson International Airport, which is the most international airport in North America at the moment, would be an ideal way. And in fact, we did that. In fact, I should say, not we did that, we are doing that. It's likely the program will end in March, but we are testing at Pearson Airport currently for SARS-CoV-2 variants as they enter. We've been doing this for 3 years now. And then other things that my lab has been doing on the right side is testing in the city. So we were part of the provincial wastewater testing initiative. We'll talk about it in more detail later. Where as part of that, we were taking samples from Guelph, and in our case, Niagara region. There were 13 universities, and each university had a different regions. So ours was Guelph and surrounding area, and also Niagara region, Niagara Falls, and Fort Erie, and places like that. And we would take the wastewater and test it and report that to the province. That program ended at the end of September, which is, for all the reasons I've told you about, is a tragedy. We've been able to scrape together some funding to continue it again in Guelph, and are trying to find funding to get it up for most of the province again. And our results are shown and are placed on our local public health unit, the Wellington Dufferin Guelph Public Health Unit website, so the public can go and see. So this is real-time data. It's updated weekly. Um, and so what you can see is the graph on the right is more recent. So what you can see, so we're testing for 3 diseases. We're testing for COVID, we're testing for influenza, which causes the flu, and we're testing for RSV. These are all major respiratory diseases that cause illness. And so you can see, if we look at the graph on the right, that RSV is way up. RSV tends to cause illnesses in children, so children in schools and so forth. Certainly, our kids have been in and out of school with respiratory illnesses, and many kids have been, likely RSV. But we can also see that influenza is up, influenza A, which is the main influenza. And we see that COVID, the purple line, near the end of the year, there was a increase. So that's like kind of halfway between October and January. At the bottom there, you can see an increase, likely around the holiday season when people congregate. But then it's come back down, and it seems to be decreasing at this moment, which is good news. So this data is publicly available. Again, this is one way we can address health inequality. Anybody can go to this website in Guelph and look at this data and then make decisions about their own self. Maybe if they don't want to go and congregate in a high congregate setting, maybe they want to wear a mask, um, as much as possible. Uh, given what I said, that marginalized people tend to work in jobs where they can't necessarily or don't have the freedom, to quarantine or self-isolate and so forth. So that being the caveat, at least people can see what the situation is and try to reduce the risk. So let's move on and talk about foodborne diseases now. Um, this is what I do mainly. I study foodborne disease. Why? Because it's a major global problem. Uh, this is data that's outdated. It's about 9 years outdated now. But almost at that time, almost 1 in 10 people, which at that time was 600 million people across the province, or sorry, across the world, would fall ill due to a foodborne disease, which led to over 33 million healthy life years lost and 420,000 deaths, with a third of those deaths occurring in children under 5. And while we like to think of waterborne and foodborne diseases as things that happen in faraway lands, like in developing countries, low and middle income countries, um, we know that's not true. Anybody who's been paying attention to the news in Canada and the US will have seen a large number of foodborne outbreaks this year. Um, with respect to waterborne outbreaks, we know there's still large numbers of indigenous communities that don't have access to clean water, and so that's a source of disease. So we have these problems right here in Canada. And so we know that every year there's about 4 million cases of illness, which works out to about 1 in 10 Canadians. And we if the foodborne illness, and we know these are caused by 30 different bacteria, and this leads to 11,600 hospitalizations and 238 deaths. And again, at the bottom there, those are just two outbreaks. Recent outbreaks have occurred here in Ontario, but it's not exhaustive. And then, as I said, when we were talking about the pyramid of surveillance, the problem is we don't know the true numbers of illness because most people don't seek help. So we have multipliers that can help us to estimate, but we don't know the true number. And this is a study from 10 years ago, 11 years ago, showing that in Ontario, um, only 4% of the estimated true number of cases were reported. Only 4%. Okay, so we don't know about 96% of the cases, and that's really important because certainly with foodborne illnesses, which were thought to be mild, we know that they can now lead to chronic illnesses, which can actually uh, cause death. And so in 2020, uh, just as the pandemic was started, I received a large grant from a funding agency called Genome Canada. It's a $6.3 million grant, ends later this year, to actually develop with the Public Health Agency of Canada an approach to use wastewater-based epidemiology to try to identify foodborne outbreaks faster. And we actually combined it with social media syndromic surveillance. So social media syndromic surveillance refers to looking on social media for people talking about symptoms associated with disease. While people will not go to the hospital to report illnesses, they often talk about it on social media. And so in the US, this has been used for years, about 10 years now, to track outbreaks of foodborne disease and other outbreaks um, of illness. Some of you might remember something called Google Flu, which is no longer around, but it's an online program that would also track illnesses and so forth. And so we combine it with wastewater because wastewater can only tell us at a population level that there's a signal, right? So we're sampling in Guelph or maybe Toronto, and we see a signal. So we know that there's a signal, but we don't know who's infected. And really, we need to know who's infected because that's how public health officials find out the source of the outbreak. They investigate, talk to the sick people, find out what they ate, and try and get that away from the grocery store so less people get sick. And so we think that if here's our pyramid of surveillance, we think that if we initiate this approach, we'd go from 3 to 4 weeks of reporting down to 1 to 2 weeks. And that could significantly reduce the amount of people sick by instituting this active wastewater-based surveillance method, which would be at the bottom of the, when the population is exposed, as opposed to where the surveillance traditionally is, which is at the top of the pyramid once it's reported. So we look for a number of foodborne pathogens. Now I told you there's 30 different agents that are monitored. So we look for common cases. Salmonella, most people have heard of. Campylobacter is a bacteria found in poultry, and causes actually quite a bit of illness. Sugar toxin producing E. coli. So this is one. I don't know if maybe some people would have heard of the McDonald's outbreak of E. coli that happened in the United States earlier this year. It was a lot in the news because there was worry that it would happen here, which it didn't. This was the bacteria that caused that. Listeria monocytogenes is another one. The almond milk outbreak that received a lot of fanfare is caused by Listeria. That's a particularly serious one. It doesn't cause a lot of illness, but the death rate is high, anywhere from 20 to 40%, particularly because of who it infects, which are high-risk individuals, which include children under the age of 5, the elderly, those with compromised immune systems, and pregnant women, who will, in those cases, may lose the baby, or they themselves might die. So what we did was we collected wastewater from the city of Guelph wastewater treatment plant, and it says a 26-month study is actually longer than that. We just concluded the study in December. And we use PCR, a method called quantitative real-time PCR, to measure the pathogens. So this PCR, this method, think of it during COVID, when we went to get tested, the nasal swab that they would take, they would test by PCR. It's the same approach. And this just shows how we would do it. We would go to the wastewater treatment plant, we collect the wastewater, we have to filter it, we have to extract the DNA, and then we can do PCR, real-time PCR, which tells us, as it says, in real time. You can actually see if the pathogen is present. And we can also determine the DNA sequence, which is useful, as you'll see later. And so here we see the results. I don't want to spend too much time on this. But you see in the top left is salmonella, and the top right is the Shiga toxin producing E. coli, the E. coli that caused the outbreak in the McDonald's hamburgers. And the bottom left is Listeria, the bacteria that caused the outbreak in the almond milk. And then on the bottom right is Campylobacter jejuni, the bacteria found in poultry. And the lines, the black lines, that represents cases of people. So the cases per 100,000 of the population in Guelph. That we obtained from the public health unit. And then the dots are samples, wastewater samples that yielded the agent we were testing for. So let's focus on salmonella at the top left there. You can see the dots. There are times when, and then the dotted lines show the limit of detection of the test, the baseline, and what we've defined as an illness threshold, above which we think if we got a signal, there would be definitive cases of illness. And so you can see that there are times we found signals in the wastewater. Now, one of the things about wastewater-based epidemiology, a great thing is it's actually often a leading indicator of illness. That is, you can see the pathogen in the wastewater before people get sick. During COVID-19, this was shown continually, where we would see signals about 14 days before, in some cases a month before, there was a large number of illnesses. And that would be really useful for several reasons, because we could tell people of that, so people could start to quarantine. Also because we could work with hospitals and say, well, it looks like in 2 weeks or so, you're going to have a large number of illnesses. So maybe you want to think about ordering more PPE. Maybe you want to think about developing, call-in lists for nurses and doctors and other hospital employees, which we know is a problem during the pandemic and so forth. So when we look at this here, we can see that we do see increases in the wastewater prior to some increases in the number of cases. Remember, that's the black line, but not always. Not always. And we don't understand why, not always, but in some cases we certainly did. And the same can be seen for other graphs. The E. coli and the Listeria The number of cases reported during that period were very, very low. So we didn't really analyze that because the cases were too low and below the limit of detection. But the Salmonella and Campylobacter um, were, had high cases. And so there's another, when we talk about approaches to screening for pathogens, there's another way. PCR is very targeted, but we can use what's called non-targeted approaches, which will tell us everything that's there. And so we've done this. This is a method called 16S RNA gene sequencing, which basically tells us all the bacteria that are present in the wastewater. It's more expensive. It takes longer than PCR, so we wouldn't use it routinely. But what we did here, I just put this up here to show you some of the other foodborne pathogens that are present in the wastewater. Everybody will likely know about Clostridium botulinum. If you've heard about Botox, this is the bacteria that produces a toxin, the most the most the most to- the most potent biological toxin known to humankind. And so this is where if you eat that, and every year, there's certainly some people who get sick. For example, if you can foods at home and you don't can the food properly, like in a pressure cooker, this bacteria can grow and produce a toxin. If you eat that and you do not seek medical care, you will die. And every year there's about one or two people, maybe three people that get it, and usually they recover because they go to the hospital. You have to get an antitoxin, but if not, what happens is a toxin. What it does is it paralyzes your muscles, and so people die of suffocation because when we breathe, we require our lungs to contract, and if they're paralyzed, they can't. So you die of suffocation. So this is why Botox is used. Right? Botox, small injections into the face. What does it do? It removes wrinkles. Why? Basically, there's a local paralysis of your face where the muscles are with the wrinkles, so the wrinkles disappear. Other pathogens, Vibrio, very common one in water, and so forth. So my lab really focuses though on Salmonella, and the reason we do is it's a major global issue. You can see the number of illnesses in the world each year, 93 million, 150,000 deaths. In Canada, about 88,000 cases and 900 deaths. And you can see in the US. Major issue um, in developed countries. It's even been thought that it led to the decline of populations, such as the Aztecs historically. So very interested in that. Also because it causes an outbreak or it can contaminate virtually any food. Okay, and has caused an outbreak in virtually any food. So this slide I put up these are all foods where salmonella has caused outbreaks, but this is not exhaustive. Okay, so you have dry foods, you have vegetables, you have meat, you have dairy products, um, you know, chocolate, things like that. Not to scare anybody or anything like that, but I always like to do these talks. By the way, I like to do these talks after lunch and several hours before dinner, so people don't get freaked out. We do have a pretty safe food supply in Canada, but the outbreaks do happen. In cereal, you can see there. So very interested to understand how is it that salmonella can contaminate all these foods. So this slide shows just the outbreaks that have happened here in Canada due to different foods and things. Cantaloupes, corn, shell eggs, um, peaches, onions, chicken products. And then you have snakes and rodents. And so people might say, well, what does that have to do with it? Or pig ear treats, dog treats. Well, what does that have to do with it? Remember what I said about One Health. So a lot of people have companion animals, and they like dogs, which they will feed their dogs pig ear dog treats and things like that. Major source, and then the dogs can contaminate their owners. Snakes and rodents. We see all the time outbreaks linked to daycares that have pets, like frogs and rodents and snakes, and kids pet them and then don't wash their hands and touch their mouth, and they get sick. Okay, so we really need to address that. So wastewater is a great reservoir for salmonella, and so we can use wastewater-based epidemiology to track this, as you saw in the previous graph. And so, again, this just shows how we did this and what we can do with it. We can isolate. We can get the wastewater. We can isolate salmonella from the wastewater, and then we can use that to look at obtain information about that salmonella. For example, we can look at the antimicrobial resistance genes in salmonella. Salmonella is a major bacteria that's often antibiotic resistant and can lead to treatment failures in children. We can look at the genes in salmonella and link that to potential sources in the environment where the salmonella is occurring. So then we can go to the environment and try to eradicate the salmonella so that we stop future outbreaks. Again, the One Health approach. And these next two slides show that. So from the wastewater testing that we did, we isolated salmonella, and we determined the DNA sequence of them, and then we compared that sequence to other salmonella from previous outbreaks and so forth. And so that's kind of what you're seeing here. We can determine the different types of salmonella. And actually, when we compared the DNA sequence to other sequences, so this was from the Guelph wastewater treatment plant, we found one sequence in a database that was very, very, very similar, okay, in a computer database. And when we looked more closely at it, it turned out that this was a salmonella that had previously been isolated several years before from Guelph, from a farm, a pig farm, okay, and we're seeing it in the wastewater. So what that tells us now is that that pig farm could be a source of that. So let's just say that people had gotten sick, and we did an investigation, and they take a stool culture, you get the DNA from that stool culture, and it's the same. We could One of the things public health officials would like to do is say, well, let's visit that swine farm and let's see, is there any way that swine meat, pig meat, from there, is that the cause of the outbreak, and so forth. So these are all ways how we can use this to track outbreaks and try and stop them more quickly.
Lawrence, I just want to let you know you have 5 minutes left.
Oh, I only have 5 minutes left. Okay. Okay. All right. So just going on quickly, another thing we can do is look at antibiotic resistance genes. And so this is what we're doing. And the one here is this box here. These fluoroquinolones, these are the ones that are usually used to treat infections, and are is a major concern in salmonella. And so what we're seeing here is that there is some resistance to these fluoroquinolones. So that's another thing we can use that can help doctors. So let's quickly move on then, and I'll just finish off by talking about what we did with COVID-19 and wastewater surveillance. So we were part of this wastewater surveillance initiative in Ontario, which was a one-of-a-kind um, um, uh, initiative in the world. It consisted of 13 universities, and this was during the pandemic. And you can see the cities where we would sample, um, and that covered more than 75% of the Canadian population. And the data would be given to the local public health units, which would be placed on their dashboards. And you can see that there. And where did we sample from? Many places, wastewater treatment plants, but also upstream locations, including long-term care homes, university campuses and dormitories, hospitals, correctional facilities. We already talked about the airport. And so all this data was used by the public health units to provide information. And so the last thing I want to finish off on, and I'm just going to skip through some slides here, is is rapid rapid water testing technology, because this also came out of our work with wastewater. So this is a student. His name is Noah Bryan. He's currently in grade 12 at a school in Richmond Hill. He worked in my lab as a for his science fair projects for 2 years. And he was very interested in developing testing for drinking water because of the issues in low and middle income countries. And so he developed this test. It takes 9 hours. We were testing lake water because there's this lake near his house that is not supposed to be safe for recreation, but people use it anyway and they get sick. So he wanted to test to see what was in there. So he developed this test. Um, working with black students is a major passion of mine, to give them opportunities to succeed. So not only did he succeed by winning all kinds of medals at the Canada Wide Science Fair and actually going to an international competition, um, in Sweden, where he placed fifth, but he has also just been accepted into Harvard University for September. So a great indication of what we can do when we give those from marginalized populations the same um, infrastructure and opportunities as those from the majority population. And so one of the things he came up with was this rapid testing lab. It's supposed to be portable. The water concentrator that you can see is not portable. We have to redesign that, but the other things are this bento lab and this DNA sequencer. You can see the size. And the idea is that what NOAA wants to do is to develop an approach where this could be used in countries in Africa, where they have water issues, to try to find cleaner sources of water to reduce the burden of disease. And the reason we focus on Africa is because here you see this is from the World Bank and UNICEF, the percentage of global population with access to contamination-free drinking water, and the people with the lowest percentage of that live in sub-Saharan Africa, with only a third of the population having clean water. So with that, and to summarize, when we talk about health and equality, we need to strengthen rural healthcare infrastructure and community partnerships, which can be done through the use of technology, including real-time data systems and analysis methods like wastewater-based epidemiology, which can be used to monitor diseases in underdeserved regions. Um, this can also then lead to new policies uh, regarding inclusive health, and also increase funding for marginalized areas. And so with that, thank you for your time. Um, just want to acknowledge my funding and collaborations, and the other members of my team that have worked on this work. So thank you very much.
Thank you so much, Dr. Goodridge. So I have so many questions already. This was a fascinating talk, but as moderator, it's not all about me, it's about you. So I'll ask one question, and I already see that in the chat we have a couple more, and we'll sort of open it up to the rest of the folks in the Zoom room today. So one thing that came to mind from the beginning and then throughout your discussion is sort of the war the place that politics has in this work. And I thought it was really great that you actually brought up this point when you were talking about Jon Snow and how he wasn't, the government officials or whatnot didn't really quite believe him until, he had to do something to sort of twist their hands a little bit more. And I was thinking about sort of political influence in the sense that, I think what you're showing with waste management is sort of what to do, how to increase the surveillance in a way that makes more sense. But I kept thinking about like how do we actually get it from the source? And I'm thinking about the ways in which a lot of industries are deregulated and sort of farming and stuff like that, and how there's ways to perhaps find those. Like I also lived in the United States when Trump got elected, and there was a huge deregulation of farms, and there was a lot of foodborne illnesses sort of coming at that time. And while we're not quite there here, I do wonder the way in which the political sort of over neoliberalization of different industries, how that affects your work, and how, if there are initiatives that perhaps if they can't go through sort of policy or politics route or governmental route, that try to access the source of the farming, or the manufacturing, or whatever step that takes to get to our grocery stores.
Yeah, so great question. Certainly, the same things that have happened in the U.S. and likely will happen again, uh, can and likely will happen here. Um, there are certain governments, certain political parties that do not appear to endorse science and public health initiatives as well as they should. And so there's certainly a concern that could happen here. That said, with the wastewater-based analysis, one of the nice things is we collect these at wastewater treatment plants. Those are municipal in nature, so largely free from provincial or federal politics. Certainly, certainly there are some municipalities that are more uh, would endorse this more than others. We saw this during the pandemic, but I do have to say that largely by the end there, everybody was on board. We continue to get access as needed. Um, it's not. I mean, wastewater treatment plants have to test their wastewater to make sure that their processes are working anyways. So we just ask for another sample, and they're usually willing to give it to us. The issue becomes more of an issue when we start to talk about farms and so forth. For example, bird flu is a major concern. It's infected cattle in the U.S. and other animals. When that happened in March of last year, I immediately tried to begin testing here, and was shut down because, you know, we were told by the Ministry of Agriculture that that would not be good. So um, and so because of that, largely right now, there's not a lot of testing going on. There could be bird flu here. There's limited testing. There could be bird flu. Certainly, we know that it's in birds, but there could be in cattle. We don't know because of that. So your point is well taken, and politics is always, there will always be an aspect of political um, aspect to this, and we have to try to work to find our way around it by educating them that, if we do this, it actually will lead to better health outcomes for everybody.
Thank you so much, Dr. Goodridge. So I did say that there were a few questions in the chat. There are two by Peter. So I'll start with the first one. Have you seen in Canada and the US neighborhood communities any backlash from those communities where the community members may feel that their community is being targeted? And if this is the case, how do you overcome it?
So again, that's one of the beautiful things about wastewater surveillance is that, we don't, you can do it, but we don't, and really nobody that I know of really tests based on at the community level, right? We would go and test. So there's 5 wastewater treatment plants in Toronto, and they're not stratified based on socioeconomic status. There'll be communities that are from low and medium and high in that catchment area. So all we're doing is testing that, and that really again addresses some of the historical challenges here. Groups have a distrust of the medical community because of discrimination and misuse of the data. So that's why I said earlier on we have to be very careful, when we test and make sure that we are not, affecting people's privacy. So we haven't seen a backlash. Actually, on the contrary, we've seen people help, be grateful because we're providing them with information that they would not otherwise have had.
Perfect. Thank you so much. I see we have a live question, so I'll ask this one and then I'll get to the second question in the chat. Ruth, you can go ahead.
Okay, thanks. Thanks so much for that talk. Is very, very interesting. And as Bianca was asking her question, that was one of the things that was sort of coming to mind with me in terms of you're doing all this great work. I think like your explanation of the pyramid and how it works and thinking about how what you're doing is now moving away from a response to sort of a prevention. And I guess I just wonder, we always say that we want information to be able to inform policy. And I just wonder how we move to sort of see that this is something that continues to be implemented, because I think we're sort of in this cycle of whenever we have a pandemic, we're never ready. But then it's like we have all this important work that is being done locally. And so I also just wonder in terms of if you're seeing, like, I guess, what does the message have to be? Do you think it more has to be surrounded or centered around money to show that if we are testing and able to kind of keep this sort of surveillance ongoing, that it then means it'll be less money spent for our systems? Like, what do you, I guess my question to you is, yeah, what do you think could be maybe some of the arguments to move this forward even at a municipal level? Because I think for Hamilton, as you were talking, I'm thinking this would be really great. We were one of the cities that did not have, particularly in certain communities, great uptake of COVID testing. So I just wanted to kind of hear if you had any additional thoughts on that.
Yeah, that's a great question, Ruth, and I'm going to combine that. Peter has also asked a question,
Yeah,
related to
Ruth hits on my question.
yeah. So I'm going to combine that. So before I answer that, I have to tell you, Ruth is my cousin, and I always have to do that and embarrass her because she's my cousin. So now that you know that, okay. So let me get to the question. So great question. So here, I'll give you a current example of the problem. So right now at the University of Guelph, maybe you've seen on TV, we have a major outbreak of norovirus. Norovirus is like one of those, the virus, when you hear on the cruise ships, people get sick, that's norovirus. Highly infectious, spread by person to person or through food. One residence in our campus, there's hundreds of students sick. Now, we had been testing for norovirus, but then the funding ran out, so we stopped. We stopped in November, and there's a major outbreak now, which if we had been testing, we could have maybe seen that and taken steps to try and stop it, working with public health. This is an example of what you're saying, Ruth, about surveillance is only as you have to keep continuing surveillance, including when there's not a problem. You can't wait till there's a problem, because by that point you don't need the surveillance because you already know there's a problem, and that's a big issue I see. We've seen this. We saw this in Canada. We saw it in the U.S. and elsewhere, where the problem with surveillance approaches and preventive approaches is really, if they work well, you will never know that they worked, right? Because there's no disease. So what happens is people say, well, why are we spending all this money on these surveillance methods? We've been spending money on this for 15 years. We haven't seen an outbreak. Not understanding that the reason why there's no outbreak is because the surveillance is working. So then the politicians say, well, we're going to cut that, right? So then we cut. The U.S. made major cuts to their surveillance network. Canada made major cuts to their surveillance network. So we were largely unprepared for COVID, and it's happening again. Our wastewater has the surveillance, and they've cut the funding, and so we are not ready for the next pandemic. We don't have sound controls in place. We have the technology in place. Peter in his question has made that statement. It seems like we have the technology. Absolutely, we have the technology in place, but we don't have the programs in place because there's a lack of funding. And this is the $100 million question, because politicians don't think about long term. They only think in 4-year cycles, right? Because that's the time that they're going to be in power. And so it's very hard, even though this saves millions of dollars. We can show that during COVID. We can show that. Another example, which I didn't show, a colleague of mine at the University of Ottawa, they showed that I think it's RSV. Not everywhere in the province, or it might have been influenza, but not everywhere in the province gets it at the same time. So, like, the first place that got it was like in this data was, let's say, Windsor. Toronto didn't get influenza until 2 months later. Hamilton and Toronto got it. Then, Thunder Bay gets it later, and Eastern Ontario, Ottawa gets it later. Well, that information can be used, if we really want to stagger, antimicrobials when they're bought, or antivirals when they're bought. These are types of things to save money on the public health care system. All of this stuff can be used, but there's no real political will to do it. So the real problem, the real issue, the real challenge, and this gets back to Bianca's earlier statement, is politicians. How can we have to find a way to engage with them in a meaningful way that they actually understand this and understand, but make it worth their while, kind of. So, why should we fund this? Maybe for them it's not about decreasing people. As crazy as this might sound, it might not be for them about decreasing illness, right? It might be, the economics thing is something that potentially could have some traction, although we haven't seen that yet. So if anybody can come up with a way to do this, let me know, because that's the one thing that keeps me up. If we had the money, we could be doing this and providing this info to anybody, and really reducing disease and improving health inequity.
Thank you for that. I am planning a political career in about 20 years after my professorship, so we'll see what happens.
All right, look me up. I might be in a wheelchair then, but look me up. I'll be your ally.
All right. Mohammed has a question. Mohammed, go right ahead.
Yes, well, first I want to echo the commendation, Dr. Goodridge, for the valuable and timely work that you're doing. Excellent work, and we really appreciate this work. My question is related to a slide that you showed that is about the interconnectivity of the world. You showed connections at airport, and the question is related to globalization. I wanted to know whether you've looked into the connection between globalization and the disease outbreaks that you are interested in, whether it's the foodborne illness or COVID-19. When you were talking, and particularly when you showed that slide, I thought about a colleague who is working on a paper. He titles the price globalization as the price. No, let me take that back. COVID-19 as the price for globalization. So I just want to get your thoughts about this relationship, because often we talk or we tout globalization as a good thing. People talk about the positive things that come out of the interconnection of the world. But are you seeing a relationship there that should be something we should be paying attention to as a warning, or whether the more globalization become intensified, and we'll be seeing these kinds of outbreak as an inevitable outcome of that connection?
Yeah, so wonderful question. And the simple answer is yes. So, as with everything, there's advantages and disadvantages, right? And depending on which side of the aisle you're on or what you want to do, you're going to play up the advantages and downplay the disadvantages, or vice versa. I'll start off by saying I'm a fan of globalization. I think countries as they currently exist are artificial. Like they're artificial boundaries that historically have been drawn up by those who had the power to do that. But I personally think that every person on this planet should have the ability to go everywhere else on this planet and feel free and explore that. So I'm a fan. That said, like I said, there's disadvantages. Now, one of the issues when we get to infectious diseases is that, and this comes to this One Health thing, because animals spread infectious diseases to humans. Animals have a lot of diseases that will not infect humans, because animals, their biology and physiology is different. However, humans are animals as well, and if those diseases mutate, then they can infect humans, and that's what we saw with COVID-19. And so we know that in the concern is in places where you have very high concentration of people and animals, and also which also tend to be places where there's high temperatures, like for most of the year, so that the organisms can grow and interact. That's where you see problems. So this is why we know Asia is a hotspot. China, India is expected. Africa, there's some concern there, not so much. The population's lower, but the concern in Africa is that there tends to be a lot of bushmeat that is hunted and consumed. And if you you know, if you, for example, during the slaughter process, if the blood of the whatever it is, you know, it splatters and gets onto your face and your eyes, and there's something there, you can lead to an infection. So these are the areas that are of concern. So, and then globalization happens, right? So now it's spread outwardly. Um, it is what it is. You know, again, typically when you see people who are anti-globalist, who do they tend to be? You know, they tend to be people who are already in developed countries, well-off countries, saying those people over there, we don't want you to come. Right? So it's discrimination against those who are even already discriminated, and so that's why I would say I'm against that. You know, there's going to be diseases, but this again is why we could do the monitoring at the airports. If there was a political will and money, we could do that. But even there, you saw problems. Where during COVID-19 there was discrimination. Like, so this one strain came from China, and Canada and the U.S. banned flights from China for a short period of time, which of course didn't work because how many people coming to Canada from China are taking direct flights? You can fly, you know, to somewhere else and fly it. So that didn't work. So I think we need to, and then there was another time, it was an African country, I think it was South Africa, that found a strain, and the immediate response was we need to ban, you know, people coming from there. So I don't think that's the answer. I think what we need to do is to use the technology we have, make sure there's equitable dissemination of treatments. That was another problem, right? We had all the vaccines, but in Africa and places like that, they didn't have. So make sure that there's true global you know, distribution of vaccines and treatments to decrease the health inequality. And these will be ways we can minimize the disadvantages of globalization while maximizing the advantages.
Thank you, Mohammed.
Can I ask a question?
Yes, where, Peter, yes, go ahead.
Yeah. Professor Goodridge, this was just like you've made my week and month and year. What a beautiful presentation, you know? I have so much I want to ask, but I won't. I'll make 2 or 3 quick points, and you may think the first is a bit facetious or odd, but it's incredibly important. Could you please, I'm a lawyer, and it's critically important that that young man knows they're not in any way opposed to doing the excellent social justice work he wants to do, but he should also think of his inventions in a business way as well. Okay? Just like, you know, he's going to Harvard. 3 or 4 of the folks who runs this world's business technology stuff, they quit Harvard, and they're the richest people in the world. So please let him know to think on that parallel line. And it would be so brilliant in time to see a young Black man doing such great social work, but also, you know, he needs, just if you could just let him know in 6, 7, 8 years, think about how do I go public with my corporation, you know? So
sorry to interrupt. What type of law do you do?
I do corporate governance and ethics.
So it would be great if I could get your contact info.
Okay, we'll reach out, and I would love to help him in that regard,
Yeah.
pro bono. Pro bono.
Okay.
I'm touched by this, and yeah. What I wanted to know, and I'll give you I won't mention his name. There's somebody here in North America, Oxford graduate, working on his doctorate now in law, negotiates on behalf of his country. I'm trying not to identify him. Negotiates on behalf of his country with the UN and internationally. Is a law professor, as I said, working on his doctorate in law. Getting into a law doctorate is even harder than a regular doctorate, right? And he would be in Geneva. He was telling me over Christmas last year at my home, I'd be in Geneva, and 4 or 5 taxis would pass me, right? I'm so proud of you, Dr. Goodridge, what you're doing. Have you experienced that in your world as you move around the globe?
Well, when you said that, the only one time what made me immediately think of was I was in Hawaii once and that happened.
Mm-hmm.
I spent a lot of time in the U.S. Before I went to the U.S., I was not. This whole thing of races. I mean, I knew about it, but I didn't know about it. Let's put it that way. Then I went to the U.S. And my eyes were open. And then it's changed me. Then I've come back, and people always say, "Oh, well, ooh, you're back in Canada. Well, I guess it's not as bad here." And I say, "Well, you know, depending on how you look at it, it could be actually worse here, because it's more subtle here. On the other hand, it's not as in your face, at least in my experience here, as there, where people like actually came up to me, or I'm sitting in a department seminar, and the N word comes out, and you're just sitting there as the only black person in the room, and you have to deal with that. So, so yeah, I mean, but the point is, I will say this. I think the higher you rise academically, whether it's in industry, in government, whatever you're doing, the higher you rise, the more you see it. I think that, I have this saying, and it came from my time in the U.S. And the saying is, know your place. Like, know your place, black man. Right? So as long as we know our place, there will not be any problem. And our place is where they tell us our place is. Okay? So as long as we stay in our little box, right, that they give to us, then there's no problem. They might even say, well, what's the problem? We treat you well. And they might even be, right? Like in that box. They might be treating you well, but it's still a box. And so if you dare to come out of that box, or try to come out of that and think you're going to be better, or think you're going to be more, then we have problems.
Yeah.
And that's a lesson that I've learned, and that's a lesson that I see, and continue to see. And so what it does for me is it emboldens me, and this is why with that young man Noah, I took him under my wing and will continue to. But there's others as well. I'm dedicated to having people come into my lab, black students, and I will throw the full extent of my resources behind them, and help them to succeed. It's one or two at a time, but still, um, because I think historically, because that hasn't happened, there's been so many people who have, unfortunately, could have changed the world, that did not have the opportunity to do so.
Thank you. I'll give my coordinates to Bianca and she'll forward it to you. How's that?
Wonderful. Thank you so much. And thank you for your kind comments.
Yeah.
Thank you so much, Peter. And Dr. Goodridge, for your answer. We have 2 minutes left, and if you'll indulge me, I just would like to ask one more quick question, and then we'll wrap it up. But I just, because you have such knowledge and all the data that you compiled around COVID-19 and that wastewater and the trends and the ups and down, and that you're also using that methodology for foodborne illnesses. I was wondering if you've noticed, or if there's a way of a way in which COVID-19 has sort of degraded a lot of people's immunity system, and how that perhaps makes them even more vulnerable to these foodborne illnesses, and if there's like a correlation that you've been finding through your extensive work and years in doing this work.
So I haven't done any work on that, but that's what I do know. There's a lot of concern that it's exactly what you just said, that COVID-19, there's, a lot of people have said, well, what's the big deal? The death rate, the global death rate is actually less than 1%. So that is of all the people that have actually been infected, about 8 million globally, there's less than 800,000 people have died. So people say, well, what's the big deal? And I say, well, the big deal is this is a disease that I don't think we're going to see, I don't think we'll know about the ramification of this for another 10 years. People keep getting infected over and over and over, and there's already research emerging that shows the more times you get infected, the more susceptible you become to other diseases. Long COVID is a thing, brain fog, and people can't can't get out of that, and then depression associated with that. So And I think that there's going to be problems like healthcare. We're going to have in about 10 years, I think we're going to have healthcare like a lot of people coming in with weird situations because of this disease. So, but and with respect to foodborne pathogens, I can't answer that. I don't know, but certainly one of the thoughts why we see RSV all of a sudden now and so high is because of exactly what you just said, that it could be something that COVID is depressing the immune system, and we just can't fight off these diseases. So if that's the case, then that is pretty serious. And it's another reason why we have to try to be healthy, and we have to try to prevent try to prevent as much as possible these illnesses.
Absolutely. Well, thank you so much. With no further ado, I will end this session. Please, everybody, help me in thanking Dr. Goodridge for his excellent presentation and for this great Q&A. So thank you so much. This has been great.
Thank you so much for having me.
Yes, no problem. And as we end here, I just want to let you all know that we have another Tubman Talk coming up next week, January 23rd, with Violet Ferreira Sutherland on blockchain technology and cryptocurrency as instrument of decolonization, a study of racialized community in the Caribbean and its diaspora in Canada. So make sure you check out that one next week. We also have a few more things coming up in January. February's Black History Month, so we got loads of stuff going on. So I encourage you to join us for our next events. So thank you again, and have a good evening, everyone.
Thank you.
Thank
Bye-bye.
you. Thank you.